So welcome everyone to our Kaleidoscope lecture. I'm very excited to hear the work of Dr. Jeff Hovis. First, I would like to start with our land acknowledgement. The CRSC's activities related to sharing color knowledge take place across Canada, understood as part of Turtle Island, the ancestral homelands of over 630 First Nation communities, representing more than 50 Indigenous nations and languages. We work to respect, affirm, and support where necessary the inherent treaty rights of all Indigenous peoples across these lands and waters. Our commitment to honoring the knowledge, ways of knowing, and wisdom of Turtle Island's Indigenous peoples aims to contribute to the decolonizing work required today for moving towards a just society. So welcome. I'm Robin Kingsborough, president of the CRSC, and this is our Kaleidoscope lecture series. I would just like to do a quick announcement before we get going. We actually have a membership drive on now. And if you become a member before November 15th, you are actually automatically entered into a draw to win a book. Kai Cooper-Schmidt's Blue in Search of Nature's Rarest Color, and we're going to have members night book club coming up in December, so just in time to read the book in advance of the book club. So if you are not a member, please consider joining, and these are some of our membership benefits. They're listed also on our webpage, so I'm not going to go through them right now, but at the bottom of our webpage you can scroll down. Or if you know someone that you think enjoys color and would like to become part of our community, we would love to have them. So I'm very happy to welcome Jeff Hovis, and I call upon Venkat, our treasurer, to do the introduction. Thank you, Venkat.
Thanks, Robin. Good evening, everyone. Welcome to our Kaleidoscope lecture. It's my pleasure to introduce today's speaker, Professor Jeff Hovis. Dr. Jeff Hovis recently retired as a professor at University of Waterloo School of Optometry and Vision Science after working there for 38 years. I've never stayed in one place for more than 5 years. So this is a great achievement. I don't know. Okay. His research is on visual performance and color vision contributed immensely towards fundamental understanding and in developing various vision standards in aviation, rail, and maritime transportation, along with law enforcement. Dr. Hovis has also been investigating visual performance and optical properties of ballistic protective eyewear for Canadian land forces. Jeff received his Doctor of Optometry and Master of Science degrees from Ohio State University. He then completed his PhD degree in physiological optics from Indiana University. He has been a member of CNC CIE, that's our partner organization, the International Commission on Illuminations, Canadian National Committee, since 2015. He was a past president there. And now, he's part of the CIE Division 1 Vision and Color member. Jeff has a special interest in how effectively individuals with color vision deficiencies can interpret color information. So today's talk by Dr. Jeff Hovis will share his research insights on how low oxygen environments affect the basic color discrimination, with a special focus on individuals with congenital color vision defects. Welcome, Jeff.
Thank you very much. Let me see if I can get my slides up. It worked previously, so I'm optimistic. And let me...
That looks good.
Yeah, there's one other little thing I want to get. Here we go. Got the laser pointer. Okay, very good. Thank you. Thank you very much for the invitation, and Venkat, the nice introduction. Not too many people say nice things about me, so it's always good to hear that. Today's talk is going to be on color vision deficiencies and hypoxic environments, kind of a catch-all of some research I've been doing over the years. The first part was done in conjunction with Nelda Milburn and Thomas Nuthouse at the Civil Aviation Aerospace Medical Institute in Oklahoma City, which is part of the FAA. And let's see if we can get, okay, there we go. Have to use a key. As such, the funding for this came from the FAA through various different organizations within it. And just got to let everybody know that since the FAA was funding it, the United States government doesn't assume any liability in what I'm going to say. Just to overview some color vision deficiencies in general terms, they are characterized by reduced color discrimination, which is fairly straightforward. Color matches are outside of the normal range. So, for example, the green on top and yellow in the bottom, hopefully for most people, would look identical to somebody who has a color vision deficiency, even though perhaps their color discrimination is not too bad. Or possibly they could have both these conditions. They have matches outside the normal range and also reduced color discrimination to go along with it. The color vision deficiency can be congenital, in which case it's most likely due to different types of cones within the retina or the lack of a cone type. If it's not congenital, it could be acquired through a disease or disorder of the visual system. Generally, we classify the deficiencies as red, green, or blue, yellow, meaning that they have trouble discriminating along the red-green color axis if they have a red-green color vision defect, and trouble discriminating along the blue-yellow color axis if they have a blue-yellow defect. For our purposes, we'll consider functional color vision as roughly similar for the case in congenital and acquired color vision deficiencies. There are some differences between the two. One of the ones that's probably more important is to realize that if you have an acquired color vision deficiency, you're more likely to have a difference between two eyes in terms of your color perception. And also with the acquired color vision deficiencies, you tend to get overall reduction with a larger reduction in one of the along the color axes. We'll start off talking about the congenital types because the acquired ones can loosely be fit into one of these categories. A protan color vision defect has someone who has difficulty discriminating between the reds and greens or the red-green color axis. The problem that they have is with the red cone, but we don't like to use the word red since the cones themselves don't color. So it's actually more commonly referred to as the L cone or long wavelength sensitive cone. They could either have a different sort of L cone compared to the color normal population, or it could be lacking. This is about 2% of the Caucasian males, which reflects the fact that it's X-linked recessive trait. And so females, it's relatively rare in them. In terms of some aviation displays, we have a navigation display, which is basically a compass and different directions identified by different colored arrows that would be seen by somebody with normal color vision. And in a simulation of protan vision of somebody who has a fairly severe defect, that would appear something like this. I'm always a little bit leery about showing these pictures because I don't really know what a person with a protan defect sees. But one thing it does highlight is what colors they're likely to get confused. So the arrows, which are pink and blue here, are almost the same color for a person who has a protan defect. You can see that the green versus the orange, quite obvious difference there. But for a person with a protan defect, it's more subtle. I mean, obviously there's a difference there in terms of the saturation or the vividness, but it's not as distinct as it was over for a person with normal color vision. The second type that's also a red-green color vision defect is a deutan, and this is a person who has a problem with the green cone or the M cone or the medium wavelength sensitive cone. Again, it could be different or it is lacking. This is the most common type of red-green color vision defect. About 6% of the Caucasian males have this, and a little bit higher, but very rare in females compared to the protan defect. And if you look at the navigation display for the simulated deutan defect, the severe one, looks very similar to what the protan did. What's the difference is these two arrows are perhaps a little more distinct in difference in color, and these two arrows are more similar in color than perhaps the protan. But generally speaking, in terms for this display, they see colors or have trouble with colors in a similar degree. There are some differences between the two, but out in the real world, it's somewhat more difficult to tell apart compared to the laboratory. And the third type is a tritan defect, which is a person who has difficulty distinguishing along the blue-yellow axis. It's technically a violet-yellow-green axis, but violet and yellow-green is hard to say. So people tend to call it a blue-yellow discrimination loss. This is a problem with the blue cones or S cones. They're not functioning properly. The locus for this defect for the congenital type is on an autosomal part of the chromosome, so it affects males and females equally. However, it is an exceedingly rare color vision defect in the young adult population, with less than 0.1% of the people affected. As you get older and problems start with the visual system, then more and more people have a acquired tritan type defect, which in some populations can be as high as 60% of the older population. In terms of hypoxia, you start running into trouble with different aspects of functioning, whether it's vision or cognitive, when your hemoglobin oxygen saturation gets approximately 90% or lower. We tend to refer to this as blood oxygen saturation, and it is basically the percentage of saturation of the arterial hemoglobin, the SaO2, which is the ratio of the oxyhemoglobin molecules to the total number of hemoglobin molecules that are available in your arterial blood supply. At sea level, your saturated oxygen level is 98%, and when you get up to 10,000 feet or about 300,000 meters, it drops down to 92%, which means that you're going to start running into trouble. Just wanted to remind you that the percent of oxygen in the atmosphere is the same at all altitudes. What the issue is the air pressure decreases, and so that sort of spreads out the molecules quite a bit in the atmosphere. And so the number of molecules per breath that you can inhale actually decreases. And that's why you run into problems, is you're not inhaling as many molecules of oxygen at the higher altitudes compared to sea level. In general terms, below 5,000 feet or 1,500 meters, hypoxia is not a concern unless you have a health problem of some sort. Above 7,000 feet or 2,000 meters, then things start to change. Breathing and heart rates start to increase. Coordination can become impaired. Fatigue becomes an issue. And if you have a rapid exposure to altitudes over 8,000 feet or 2,500 meters, then you increase the risk of developing altitude sickness. In terms of some of the cognitive or perceptual effects that happen with hypoxia, around 5,000 feet, you start running into trouble with seeing in the dark. And this is basically out in the woods alone with no flashlight and just a quarter moon type of light levels. When you get around 8,000 feet, your short and long-term memory can be affected. You start running into problems with color discrimination. It becomes impaired. And you have also reduced sensitivity to contrast at lower light levels. 10,000 feet, you're starting to run into problems with hand-eye coordination, particularly the more complex ones. Performance on conceptual reasoning tasks decreases a bit. and you get some minor reduction in color discrimination at brighter light levels along the red-green axis, actually, instead of the blue-yellow. Then you get up to 12,000 feet or 3,600 meters. The performance of pursuit motor tasks degrades. Your reaction times lost an eye there. Reaction times increase. You end up with a bit of tunnel vision, and your pupils start to constrict, which means you're getting even less light to the eyes than you were at sea level. At 15,000 feet, you can develop fine hand tremors, which is going to make precise adjustments more challenging. And about 16,000 feet, you're going to get more severe color discrimination losses, primarily along the blue-yellow axis. And also you start running into decreased sensitivity to flickering lights. Why this is an issue is that pilots in the US are permitted to fly up to altitudes of 12,500 feet without supplemental oxygen. Oddly, most other jurisdictions set the limit at 10,000 feet. Not too sure why the difference exists, but it does. And this study was basically prompted by the National Transportation Safety Board in the US to investigate whether individuals with congenital color vision defects have a greater impact in color discrimination or loss in color discrimination in the mild hypoxic environments. Previous studies showed some minor changes in color discrimination in these individuals, but there was only 1 or 2 subjects that the data was collected on. I was fortunate to work with Nelda and Tom on this project, and we conducted the study in the altitude chamber at Oklahoma City. The first part was to do everything at ground level, which was about 1,000 feet above sea level. The session for all the testing lasted about 4.5 hours. Basically, if somebody is going to use the altitude chamber, they're going to do as much testing and research as they possibly can. So multiple people were involved in this study besides us. At altitude, the heart rate and SaO2 was monitored using a finger clip-on. And the subjects, we had 14 people with normal color vision and 7 individuals with a severe color vision defect. We'll call them dichromats, and 8 individuals with less severe color vision defects. We'll call them anomalous trichromats. Individuals with normal color vision are called trichromats too, but they're called normal. And all 15 of these individuals had 1 of the 2 red-green color vision defects that we talked about earlier. The tests we used were a combination of clinical tests / research tests and also of practical tests that we use. The first one is the Rabin cone contrast test, where a colored letter is presented within a gray background. And if you identify the letter correctly, then it becomes more pastel and it keeps fading into the gray background until you no longer see it. And the contrast of the letter that you can just barely see against the gray background in terms of the color is what's recorded. The CAD test is a little more interesting, I guess, to say. You see the colored square that's here, and this is actually moving from one of the corners going from down to up, or up to down in one of the 4 diagonal directions. And in addition to the movement, these black and gray dots, black, gray, and white dots are alternating in time. So you have a scintillating background of gray dots with this colored object moving along the diagonal. If you can identify the direction correctly, then it gets harder to see by the reducing the color contrast or making it more pastel until you can no longer see it. And then they find out what's the minimum color contrast or saturation, if you will, that can be detected, the motion can be detected. On the more practical side, we had the Holmes-Wright Type A lantern. This was used for testing color vision in aviation candidates, primarily in the UK, but it was used in Canada for a while. It presents two lights. You can see a green, sort of pale green here and a red down below. These lights are the ones that are used on aircraft, the green, white, and red lights for navigation and running lights that are seen on the outside of other aircraft. Two lights are always presented, and the task is to identify the color of each light individually. And the results were somewhat disappointing, in the sense that we had very minor differences in performance and color discrimination. So in terms of the Rabin cone contrast test, we had three colors, the pink, green, and magenta, that were used. The pink is designed to stimulate only the L cone. The green stimulates only the M cone, and the magenta only the S cone. So these provide an index of each of the individual three cone pathways. Values near the 1 line or the 0 line indicate that there's very little change, that there's no change relative to the ground altitude. If they're above, then there's an increase in performance. They got better. And below the 0 line, they got worse. The dichromats, the people with the more severe defect, showed actually a little bit of an increase in ability to detect the pink letters at altitude, probably a practice effect, not too sure. And then about the same at altitude as ground for the green letter, and the magenta letter marginally worse. Both the normal trichromats and the anomalous trichromats didn't show much of a change for the pink letter or the L cone. Slight decrease for the green, and about the same as ground for the magenta, which is tapping the S cone. So if anything, probably a slight decrease in sensitivity or ability to see green colors against a gray background. And this represents about a 15% change from ground in terms of a decrement. On the CAD test, in terms of being able to see the different colors along the red-green axis or the blue-yellow, one indicates that there's been no change relative to ground. Values higher than 1 indicate that there's been a decrement in performance, so they needed a larger contrast in order to see the motion. And below the 1 line means they actually had an improvement. So for the people with normal color vision, there was actually an increase in threshold or an inability to see the target along the red-green dimension for the normal people. And for the blue-yellow, it didn't make any change. The people with a milder color vision defect also showed an increase in threshold or harder to see the motion along the red-green dimension. Not so much for the blue-yellow again. And the most severe color vision defect people didn't show any change in the red-green and did show an increase in threshold, or it was harder to see the motion for targets that are in the blue-yellow discrimination direction. So the question is then, was the decrement in performance related to saturated oxygen levels? Because that's one of the issues that happens when people go up in the altitude chambers, is depending on how well they adapt, their saturated oxygen levels may or may not change. And so we have the relative change in thresholds for the CAD, for the blue-yellow color discriminations. And the 1 line indicates no change. Above the line indicates that they got worse at altitude, and below the line, they got a little bit better at altitude. And surprisingly, what happens is that if you have less oxygen in your blood, you actually get marginally better. which we weren't quite well seeing that, particularly for the blue-yellow colors. And then for the red-green dimension, there really wasn't any trend in terms of changes in threshold or ability to see the targets as a function of oxygen-aided blood levels, although you can see that a number of normals and a number of people with anomalous trichromatic vision tend to have an increase in threshold at altitude, regardless of what their oxygen levels were in their blood. So how to explain what was going on is going to require a little bit of background information and a little more detail about what's going on with the test. For the CAD test, the background, as you may recall, is a gray, which is represented here. And in the coordinate system, if you're not familiar with it, this is a CIE XYZ space where X is basically a red dimension and Y is a green dimension. Blue is coming out at you. Going from the gray to this direction takes you into the oranges, reds, and reddish purples. Going in this direction takes you into the magenta colors. Going in this direction takes you into the blue greens, greens, and going in this direction is the yellow greens. The lines that you see here are colors that were actually presented within the CAD. So they have a bunch of colors in here and here, and also in the red-green area. And the ellipse that you see forming around it is basically a discrimination ellipse, whereas everything within the ellipse would appear identical to the gray, and everything at the ellipse or beyond would be visible relative to the gray for people. And this is fairly typical in terms of what a discrimination ellipse looks like for this particular test. It's longer, more distance between along the blue-yellow axis, the tri-10 axis, than it is on the red-green, meaning that your color discrimination in general is not as good along the blue-yellow as it is on the red-green. And what we think is going on in terms of our study is the discrimination ellipse itself is not changing size per se, but it's actually rotating. So instead of having more trouble exactly on the blue-yellow axis, which is here, it changes to this direction, more vertical. And so this results in a decrease, relatively speaking, in your blue-yellow thresholds with a slight increase in the red-green thresholds. And the graph over here shows the percent oxygenated blood and how much the ellipse has rotated. And you can see that there is a correlation. It's not a good one, but there's something there. That as the oxygen levels go down, there's more of a rotation to the ellipse in the counterclockwise direction. So not a change in size, a change in orientation in terms of what colors people get mixed up or have trouble seeing. Well, that's all and good. The question is, though, is there any functional impact in terms of these changes that are, well, relatively subtle in my opinion? And so we looked at the Holmes Right Lantern data. The black bars on here represent the percent of color normals that pass the test, and the gold bars represent the percent of people with a color vision defect, regardless of severity, that pass the test. The first thing you'll notice is that the Holmes Right Lantern is actually quite challenging for a person with a color vision defect in that only 10% of them were able to get within passing performance. And then for the normals, almost, but not everybody, passed at ground. When we went up to the altitude in the chamber, the color normal who failed at ground actually passed. So we had 100% pass rate for people with normal color vision at the 12,400 feet level. But the pass rate for the people with color vision defect did not change. This actually is not too surprising, but the good news is that the people who passed down here also passed at the altitude. So that meaning that, yes, it is a challenging test, but if you can pass it at ground, you're probably going to pass it at altitude. Then in terms of the number of or percentage of errors and were made on the different lights at the two different altitudes, the red represents the percent errors on a red light. So they called it either green or white. Most commonly, a red light was called white rather than green. Errors increase on the green lights. They tend to get those wrong about 40% of the time. It can either be red or white, with white being the most common error. And white, the errors are the most numerous. Again, these can be either green or red error, called green or red. Most commonly, though, people get the white mixed up with the green. And overall, the total errors are around 35% on the lantern test, whereas the percent errors for people with normal color vision would be 0. At altitude, there was a slight change in the number of green errors that occurred, but statistically, there was no difference in the errors at ground and altitude for the people with a color vision defect. And the people with normal color vision did not make any errors on the test. So functionally, there doesn't look to be a big difference between color vision for people with normal color vision or for defective color vision. In other words, people who have a mild one who can do the lantern test can still do it at altitude. So what we have here, this part is we have an increase in the red-green thresholds and a paradoxical decrease in the blue-yellow thresholds as saturated oxygen decreases. We think this is due to a change in the orientation of the CAD discrimination ellipse. This is not new data. We actually see it in data in Brown's discrimination experiment in 1951 and Jennings and Barber in 2010. They were at ground altitudes, but they found that this counterclockwise rotation occurred when you decrease luminance from indoor light levels to sort of dusk type or just around sunset light levels. And if you decrease the luminance further, then the ellipse actually starts enlarging, and particularly along the blue, yellow, or tritan dimension. So we think this gives support to the idea that hypoxia is equivalent to wearing or reducing the light levels coming into your eye, and we'll talk about that a little bit more. If you have a fairly severe color vision defect, dichromatic, you only have 2 cones represented in your retina, then basically you're only going to show up a blue-yellow defect. The equipment that we had just wasn't good enough to measure thresholds for dichromats along the red-green dimension. We ran out of contrast on the colors. From a practical standpoint, if you have large color differences and you can discriminate them at ground, which would be more operationally related, there's a highly probable that you will pass at altitude, meaning that these changes that we've seen for threshold type or just noticeable difference type experiments are not big enough to affect your functional performance in terms of detecting running lights or navigation lights on aircraft, which is relatively challenging. And the idea that this counterclockwise rotation is occurring and it parallels what happens when you decrease the light levels supports a hypothesis proposed by Conley a few years ago that hypoxia is analogous to viewing the targets or the world through gray tinted sunglasses. And roughly speaking, our data suggests that it could be equivalent to sunglasses that transmit 50% of the light. Typical sunglasses transmit about 20%, so it's not quite as dark as typical sunglasses. We're not too sure what mechanism is responsible for the change in the threshold. Hypoxia, as we mentioned before, does produce pupil constriction, but it's not enough to reduce the light levels by 50%. It's only around 10%. It's possible, based on other experiments looking at animal models, that the hypoxia occurs affects visual processing very early, perhaps where the photoreceptors synapse with the next layer of retinal cells, if not later on within the visual system. It certainly could happen as early as that first synapse between the receptors and the next layer up. And the changes at 12,000 feet that we see in this thresholds, both for color defectives and color normals, are not likely to be operationally important, providing you're at relatively bright light levels. So we're going to switch a bit and talk about cognitive effects. And the task is to say the word that's on the next slide. And then the next task is to identify what color, to name the color of the letters on the next slide. And the third task is to identify the color of the word, which is red. So a certain percentage of individuals will call this, when I say that question, will actually read blue, and everybody will be a bit slower in giving a response to this target compared to that target. So many of you may be aware of it. This is the Stroop effect. The automated neurophysiological assessment metric is basically the computer package that we use to assess the Stroop effect. Basically, this is assessing the speed, selective attention, and interference that occur at cognitive levels. There's three conditions. The subset one is basically identify or read the color, red, green, or blue. You enter the response, red, green, or blue, corresponding to the actual name. Two is identify the color of the X's, so red, green, or blue. And the third task is to identify the color of the letters in the word, which are green, blue, and red, which even reading the slide is a bit challenging. And so this process is looking particularly at cognitive functions, frontal lobe type processing, executive type processing within your brain. The reason that this was done is there were at ground level, wraparound showed that individuals with a color vision deficiency tended to be slower to respond to all stimulus categories using the Stroop experiment. And they used red, green, and blue in their experiment, which turned out to be the ones that we used by chance. But he found the interference effect, which we'll talk about in a little bit. This is a case where the letters are a different color than the color name. Were similar for the people with normal color vision and individuals with defective color vision. So the results turned out to be a bit unclear at first. And so we ended up separating things in terms of age, because that sort of gave us a little bit better picture about what's going on. And we'll start with the ground results averaged across all subsets. So one of the things that came out of this was older folks tended to take longer, and longer is measured in milliseconds. So this would be the 1 second line. And the younger people obviously are a little bit faster. So that's good, makes sense. What we were surprised to find was a slight interaction between people with normal color vision and young tended to do worse overall than people who were young and had a color vision defect. Whereas older folks who had a color vision defect tended to do worse uh, than their age group across all subsets. When we went up to altitude, um, things changed a little bit. But to tell you first about the lines, this solid line is basically the data from ground. And the gray dashed line is also the data from ground for the older folks. So in terms of the altitude effects, the reaction times for the older folks didn't change or got a little bit better if you had a color vision defect and were older. If you were younger and you had normal color vision at altitude, you got worse, basically became old in a way in terms of your reaction time. But if you had defective color vision, you didn't change. You stayed about the same as you were at ground level. And then there was a post-ground, meaning that at the end, after they returned the altitude chamber back to sea level, they redid the test again. And the people who were old with normal color vision stayed about the same as they were at ground before things started. The people with a color vision defect who were old tended to do a little bit better than they did at ground. And the younger folks didn't change relative to ground. So nothing really kind of made sense here other than to say that, well, if you have normal color vision, you're probably going to do worse than somebody who has defective color vision on overall in terms of the reaction tests, which kind of didn't make sense, but that's the way our data turned out. When we looked across the different subsets, so this would be identifying the word in black. Again, if you're old, you're going to be a little bit slower. If you have a color vision defect, and you're young, you're going to be a little bit faster than people in your same age group. So generally speaking, what we saw before was probably just due to the fact that this particular group of people, the young individuals that had a congenital color vision defect, were just generally faster, period, at responding, regardless. And then when you went to the different colors, you might expect individuals with a color vision defect to be a little bit slower to respond. And that was not the case. In fact, they actually did a little bit faster, relatively speaking, if you were older, not much. And if you were younger and had a color vision defect, it didn't make any difference. And if you're older and had normal color vision, you actually did a little bit faster, probably indicating that there was a practice effect going on here. And then when we went to the incongruent case where the letter color was different from the word, things got a little bit longer to do. So again, this line represents the ground level. Sorry, this represents the identifying the color, identifying the word, and the word letters are going to be in black. So both the color defective groups that were young and the normal color individuals with young took them a bit longer. So from 700 milliseconds up to close to 1,000. For people who were older, they also showed an increase in the reaction time, with the people with a color vision defect showing less of an effect than persons with normal color vision. So again, that's the line for the just identifying the word. So generally, people got worse, which is what's expected when the letters are different color than the word. However, in general terms, the people with a color vision defect tended to do a little bit better, or not, probably to say didn't do as bad as people with normal color vision. So they weren't generally slower. The interference is a way to look at the data, and that's simply the difference in reaction time between subset 2, which is the color of the X's, identify what color they are, versus the incongruent case where the letters and the word are incongruent. And so the difference in reaction times to those, not surprisingly, again, older folks had a larger difference. But if you had a color vision defect, you tended to have less interference. So what we mentioned before, it shows it a little bit better. At ground, similarly at altitude, although things did shift up a little bit for the people with a color vision defect, and down a little bit for the older folks. And then when you got back to sea level or ground level again, kind of the younger folks tended to do worse, have more interference than the older folks. But the same trend is case, the color vision defective people tended to do a little bit better in the sense that they had less interference in this particular case. So that's, again, not consistent with the other results that we saw in an earlier study. The fact that the young folks showed a worse performance at after returning to ground is likely due to a fatigue factor. And it does suggest that what we saw at altitude might not be directly related to hypoxia effects on the brain and elsewhere in the body, but actually due simple to fatigue that accompanies hypoxia. So that's something to keep in mind. Now, one of the reasons that we might have gotten different results from Graperon in terms of the interference effect and the color defectives not being slower in general is the red, green, and blue colors in their experiment were dimmer. So things are going to be slower then to begin with. And also the green and the red were actually harder to tell apart for a person with a color vision defect compared to the green and the red that were used in our study. So I guess the moral of the story is that if the colors, even though they might be on the red-green dimension, are present with the Stroop effect, if they're relatively easy to distinguish for a person with a color vision defect anyway, you're not going to see any decrement in performance. So the idea that the individual with a color vision defect could have less interference suggests that they may be more amenable to or show a larger practice effect. Of course, you can't call that practice in Stroop effect, but it's called a list level congruency. Basically, what happened was the colors were present in sequence instead of in random order. So that does give some practice effect and makes it a little bit easier for the subjects. And particularly individuals with a color vision defect might benefit from that. The mild hypoxia only marginally increases the interference effect. And so there's only marginal changes in executive function. This is similar to what other folks have shown. And the post-altitude data for our younger subjects suggests that any increase in interference at altitude could be due to fatigue and not that's produced by hypoxia and not directly related to hypoxia on the executive function centers or the frontal lobe. Now we're going to change gears a little bit and talk about extreme hypoxia. This sort of happened by chance. I was not on this expedition, so Harvey Langford was. He contacted me out of the blue and had some questions about color vision and hypoxia, and then things exploded from there in terms of our collaboration. And he was it's a very, like Nelda and Tom's, a very fun person to work with, and his historical perspective on mountaineering and mountain expeditions is immense, which is not good for me because I'm easily distracted. So he and a few of his friends decided that they would go mountain climbing in the Himalayas, particularly the Murrah Peak that's in Nepal. The expedition that we're talking about, they took 9 days to reach 5,400 meters, and they camped at an area that's called Murrah La before ascending further. The 9 days is sort of a typical type of duration required to go up to that height at the time, so you didn't get altitude sickness. So while they were camping at Murrah La and waiting to adapt to the new level of hypoxia, the group of four was playing crazy eights with cards for about an hour. It was in a bright environment, and the snow was around, although they did have their sunglasses on. And after about an hour of talking and playing crazy eights, one of them jumped up and said that, geez, he didn't say geez, but said something to that effect. The red in the red suits looks gray. And everybody stopped, looked at the cards, and universally agreed that yes, the red had gone out from the cards and everything appeared gray or black. They kind of just decided that, oh, this is just due to hypoxia, and they went on and continued to play for another hour or two. By the way, they did play Crazy Eights the night before, and they didn't experience this. So the issue is that's not typically a blue, a red-green sorry, how do I want to say it? The question is, what's the nature of the color vision defect? And probably the easiest way to start off with that. This is the original deck, a picture of that. He still had it. And this is what the deck would look like if you had a simulated Dutan color vision defect. And you can see that, yes, the red has gone out from the cards, but it doesn't necessarily appear gray to someone who would have a Dutan defect. When you look at a person with might have a protan defect, you go, well, yes, the red has definitely gone out of there. And with little imagination, you could say that, yes, the red has turned to gray in this particular scenario. And then you look over at the tritan defect, which is more typically found at those altitudes. You see that, in that simulation, the red is obviously present, and you wouldn't think that would a tritan defect would cause the cards to look gray. So we did a little bit of thinking about it, and the others done mountaineering have reported protan type of defects, so that part's not unusual. What was different, though, is that the protan defect was not present in all individuals that were on these expeditions, and the defect was actually quite subtle compared to the tritan defect that accompanied it. So this, based on the previous research, suggested that any change in the red suit's appearance would be subtle and not noticed by everybody who was playing the game, not universal agreement by any means. Now, it just so happened that Harvey contacted me in the summer, and one of the things I do when I come to work is I actually ride the bus and play solitary on my iPad. And the sun was coming up, and lo and behold, the glare off the screen actually makes the red suit of the cards on the iPad turn gray. So I said to Harvey that, gee, I don't think it's hypoxia that's causing the problem, particularly given that the protan defect's rather subtle, but I think it's more likely a glare it was the issue. So glare, as you know, it makes things harder to see, basically by reducing the luminous contrast by scattering light onto the object, and there's some also neural effects that occur. The scattering of the white light would actually decrease the saturation of the red colors and result in a more pinkish appearance of the hearts and the diamonds. But in addition to the change in the saturation of the red colors, it's also going to have that bright background off the background of the cards, and that's going to inhibit the brightness, and hue could result in the red having a black appearance. And in fact, you actually can change the appearance of a red stimulus by surrounding it by a white annulus. And you have to increase the background luminance of the white annulus about 20 times brighter than the red. And 20 times is not that big of a deal in order to change the red stimulus into black. So it's quite possible that the glare from the white background actually was, and also the surrounding snow, was causing the issue there. Equally as important is the fact that as Harvey called it, the relatively inert brains at that altitude. Recall that the group was playing cards for about an hour before they noticed any change. So they're pretty oblivious to what was going on in terms of their perception. There are reports from comparable altitudes describing cognitive difficulties in sorting, reading books, and using map or other navigational aids. And this difficulty is only partially relieved by acclimation. So in some ways, they did have the inert brains, but it also illustrates sort of the value of redundant coding, because you did have the shape to go along with the coding, and perhaps because the shape information was there, they didn't notice the change in color as much. And just to give you an idea that this is not anything new, Green, who was a medical officer in 2 Himalayan expeditions in the 1930s, one was an unsuccessful attempt to scale Everest, and the other one was successful on a peak that was lower. He stated in the book that he wrote that the special senses at great altitudes are frequently blunted, although this characteristic is not noticed by the majority of the expedition. So basically, the sensory perception can change and things become duller all the way around, but you may not notice it because your brain is inert or slow, and you miss it. So in terms of vision at altitude, I do like to think of things as equivalent to wearing progressively dark sunglasses as you go up. Around 2,000 meters or so, it's equivalent to a pair of sunglasses that transmits maybe about 50% of the light, so something that would be more on a cosmetic thing, just to take the edge off. When you get to around 5,000 meters, then the lenses are getting darker, and 7,000 meters, you're thinking that it's almost like wearing a welding filter, although because your brain is sort of inert, you may not even notice that it's on. And I thank you for your attention. So I guess this is the part where people get to ask questions.
Yeah, thanks, Jeff, for the very interesting talk. We can take a few questions now.
Jeff, do you want to stop? Yeah, share screen,
Okay.
and then people
Yeah,
can
let
cameras
me-
on if they like.
Did I stop? No, I didn't. Just hang on. Okay, there we go. Jennifer, I see a hand up.
Yeah, so that was really interesting. Something I don't really think about. And maybe this is a naive question is something I didn't fully understand, but are these effects that would also occur for ground-based occurrences of hypoxia? So if I had some kind of breathing difficulty where my blood oxygen was low, but I wasn't in a state where it was, you know, less oxygen in the air or lower pressure, would I have the same effect?
Yes, in general, the degree might not be as large. So you may have to have much lower oxygen blood levels, oxygenated blood levels than what we showed in the study. And given the subtlety of the loss in sensitivity, you probably wouldn't be able to notice it perceptually. Now you might start running into cognitive problems sooner than noticing the color perception. If you were looking at oxygenated levels in the eye, so diabetes, glaucoma, then you might definitely see something more dramatic than you would if it's a general lowering of the blood oxygenation. I
Cool.
mean, the other thing to keep in mind at 90% saturated oxygen, you are very tired and are not going to be moving around or being very active, so you may not notice anything because of your inactivity and fatigue.
Cool. Thank you.
Brian, you had a question?
I think you're muted, Brian.
Brian, we can't hear you.
You want to put your question in the chat?
Okay, probably I can ask a question in the meantime. I mean, before Brian sorts it out. Jeff, in your test, I mean, the people with color vision deficiencies, are they normal people with color vision deficiencies or professionals? Or in other words, I mean, like, are they used to performing tasks at hypoxic environments, or these are, I mean, like, just common folks with color vision deficiency?
I don't recall anybody specifically working in the altitude chambers or in hypoxia environments. They were recruited locally from Oklahoma City. So I don't think specifically, there might have been one or two, I can't say, but it wasn't anywhere the majority of them.
No, the follow-up for that was, I mean, like, do people learn, I mean, like, these things as they practice at lower color vision circumstances, or I mean, like, changing color vision circumstances?
There can be a practice effect. Probably more important is how they adapt to the hypoxic environment. You saw quite a bit of spread on the saturated oxygen levels from 75 up to like almost 95. And so that's related to the breathing rate that they have and a number of other factors. So that's more likely to be an issue than practice effects on that would occur at ground in normal pressure situations, at least in this experiment. The tests that we showed generally have fairly good repeatability, so there's not a big learning component to them.
Brian, can you? No. I think still, can you type your questions? We can read it out.
Yeah. No, it sounds like he's given up on that.
Okay.
Any other questions or general questions on color vision deficiency?
Yeah, Brian is typing, I think.
Okay. He's typing now, yes. Oh,
He's asked, yeah.
yeah. Well, basically, that's how the data comes out. They're the actual data from the program is in XYZ space. we could use whatever we wanted to. It wouldn't make any difference. It just so happened that with the sampling colors that they used, the ellipse fits the best. No, because it's a linear transformation. So it basically depends on how many colors you sample. So the cone contrast test samples only 3 colors. And so that didn't show the rotation. It just showed the decrease in green sensitivity and because it didn't sample as many colors along the blue-yellow dimension, there was no change in sensitivity. So it's more of an issue of making sure you sample enough colors to get a small change.
Okay.
Just along that line, did you try other colors other than the pretty vivid red, green, blue? Did you try less chromatic colors?
Oh, yes. In the end, what they could just barely see was very pastel. So it starts off very vivid, and then it decreases in its vividness until they can no longer see it. So we also sampled some other colors on some other different equipment, but we didn't show any effect with that. It just wasn't sensitive enough to pick it up on that particular test because it's relatively minor changes that occurred. And so I guess it's a good thing to say that if the colors are easily distinguishable for people with normal color vision and people with mild color vision defects, going up to 12,000 feet is not going to affect their performance on easily noticeable color differences.
Jeff, this is a very general question. I mean, are there any studies on color vision deficiencies on people living in high altitudes, population studies? Are they in any way different? I mean, what's are better than, I mean, people who are always living in high environments, higher altitudes. Okay.
I'm not aware of anything. It's an interesting question. There, in part, because there have been some studies that showed difference in ability to discriminate hues in general, depending whether you were near the Arctic Circle or the equator. And they related that to tried to relate it or thought it might be related to sunlight exposure and how things could change in terms of the color discrimination. But so the altitude issue would be interesting to look at.
Are there any other questions? Okay. Okay, thanks, Jeff. Thanks for a very interesting talk. I mean,
Thank you all for attending. I'm glad I was able to impart some knowledge on you, at least entertain you, and hope to see you all again sometime or again.
Yes, thank you very much, Jeff.
Okay.
Nice to see some CIE people here too. So we have our next talk is the end of November. Doreen Balabanoff will be looking at color and light in the birth environment. So that may interest you too. So just stay tuned to our website. And yeah, one more round of applause for Jeff. Thank you so much.
Thank you.
Interesting
Congratulations.
All right. Have a good evening and a good weekend, folks.
Thanks.
Thanks, Jeff.
Bye-bye.
Thanks, Jeff.
Bye.
Good night, everyone.
Bye.